Welcome back to our channel, your new home for mastering medical school lectures. Today we're discussing a very important topic in physiology: cell death, apoptosis, and necrosis. But before we begin, make sure you subscribe to the channel so you don't miss upcoming lectures. Like this video and share it with your colleagues. Alright, let's begin. What is cell death? Cell death occurs through two main processes: apoptosis, programmed or natural cell death, and necrosis, uncontrolled or accidental cell death. Understanding these processes is essential in physiology, pathology, and clinical medicine. Apoptosis is the natural, genetically controlled death of a cell. The word originally means falling leaves, symbolizing a natural and controlled process. It is often called cell suicide because the cell actively participates in its own destruction. Importantly, apoptosis usually does not cause inflammation in surrounding tissues. Functional importance of apoptosis. Apoptosis plays several critical roles. 1. Maintenance of cellular balance. Millions of new cells are produced daily. An equal number die by apoptosis. This maintains cellular homeostasis. 2. Removal of damaged cells. Cells damaged by toxins, infections, or DNA injury are safely removed. 3. Role in development. Examples include removal of excess neurons, separation of fingers and toes in the fetus, regression of certain reproductive ducts, shedding of intestinal epithelial cells, menstrual shedding of endometrial lining, and elimination of self-reactive immune cells to prevent autoimmune diseases. Activation of apoptosis. Apoptosis can be triggered in two main ways: withdrawal of positive signals. Some cells require survival factors such as growth factors, cytokines, chemical stimulants. If these signals are withdrawn, apoptosis begins. Arrival of negative signals. Examples include cellular stress, viral infection, DNA damage, increased oxidants, radiation, toxins, or chemotherapy drugs. These signals activate internal death pathways. Role of mitochondria. Mitochondria play a central role in apoptosis by releasing cytochrome C and other proteins that activate enzymes called caspases. Caspases then initiate controlled cell destruction. Changes during apoptosis. Typical changes include cell shrinkage, loss of contact with neighboring cells, chromatin condensation, DNA fragmentation, membrane bubbling, formation of apoptotic bodies, removal by phagocytes without inflammation. Abnormal apoptosis. Too much apoptosis can cause ischemic injuries, autoimmune disorders, neurodegenerative diseases. Too little apoptosis can lead to cancer and certain immune disorders. Necrosis. Necrosis is the unplanned, accidental death of cells caused by injury or disease. Unlike apoptosis, necrosis causes inflammation and tissue damage. It is sometimes called cell murder because external factors kill the cell. Causes of necrosis. Common causes include trauma, infection, lack of blood supply, radiation, toxins, extreme temperature, chemical exposure. What happens during necrosis? Key events include cell swelling, membrane rupture, leakage of cell contents, calcium overload, activation of destructive enzymes, complete breakdown of the cell. Reaction of surrounding tissues. Necrosis causes inflammation, increased blood flow, redness and heat, pain due to histamine release, migration of immune cells, edema formation, removal of debris, pus formation, eventually healing. Summary. To recap, apoptosis is controlled, beneficial, non-inflammatory cell death. Necrosis is uncontrolled, harmful, inflammatory cell death. Understanding both processes is essential for clinical medicine and pathology. Closing. In upcoming lectures, we'll continue exploring important physiology concepts. So don't forget to subscribe to the channel, like the video, share with your classmates, and keep learning with us. See you in the next lecture.